My name is Rachel Bigley, and I'm currently a student at South Eugene High School. And this summer, I had the opportunity to work with Dr. Phillip's lab at the University of Oregon, and I studied the relationship between a worm and its food. Every organism on Earth, from an ant to a whale to humans, all have a life history. Life history can include reproductive output and lifespan, and it can differ from organism to organism based on a variety of factors. These factors can include climate, availability of food, types of food, moisture levels, pathogens and diseases, and also a variety of stressors. I use Nematode as a model organism for studying these life history traits, and since we can't use humans. But what is a nematode? A nematode is a microscopic worm that ranges from 1 to 2.5 millimeters long. And in science, it's a very good model organism because its life cycle is only 4 days long, its generation time is short, and its genome has been fully sequenced and characterized. They only have 1,000 cells and 300 neurons, so it makes for a very easily maintained organism to study in a lab setting. The main species of nematodes that we used in the lab this summer was Nematode elegans, and it's hermaphroditic, which is very useful in studying fertility and longevity at the same time because it can produce and fertilize its own eggs. It's also a good model organism because it has established studies in aging, longevity, development, and fertility. We don't know much about the natural ecology of these worms, but what we do know from lab settings is their eating habits. And what they, this is a picture of its mouth, and what they do is they slurp up bacteria, and they grind it with their pharynx, which vibrates back and forth, and digest it in its gut. The main bacteria that they eat in the lab is E. coli, OP50 strain, and it's not a natural food source. But in nature, you can find both nutritious and harmful bacteria. Since we don't know much about the natural ecology of worms, there are like many limitations to studying these worms in the lab, and it's difficult to observe these worms in a natural environment because they're microscopic and they live in soil. So what we want to know is what and how nematodes eat in the wild. My main objective this summer was to look at the effects of different food sources on life history traits to gain insight on their natural ecology and observe how these worms interact with their food found in nature. The questions I want to answer are, what bacteria do worms encounter in the wild? What are the effects that different bacteria have on reproduction and lifespan? And can worms distinguish and choose between different bacteria? So my first question was, what bacteria do worms encounter in the wild? Christopher Avon, a student who currently goes to college in Florida, came to the University of Oregon this summer and was able to extract and isolate these 14 different types of bacterial species associated with Nematode. And this soil was extracted from Toronto, Ontario at the Koffler Scientific Reserve at Joker's Hill. And what we could do with these bacteria is use them with the worms in the lab setting, and that was really cool because this was all done in one summer. So we wanted to further investigate how these isolated bacteria affected the worms, and what we predicted was there would be some pathogenic worms that would kill the worms right away and not allow them to reproduce a lot and live a long time. And then we'd also predicted that there would be some nutritious bacteria that would allow them to survive and reproduce a lot and live a long time. So my second question was, what are the effects that different bacteria have on reproduction and lifespan? And what we wanted to do was we wanted to study, we wanted to observe and measure the fertility or the number of offspring worms can produce on these different bacteria and compare. And then also measure from birth to post-reproductive and then to death, or longevity, like how long they lived, and record both of those. My experiment was based on those 14 bacteria that Christopher had isolated, and with the control E. coli bacteria. And what I did was on day 1, I placed a virgin worm onto a plate. I waited a couple days, and if there was offspring on the plate, like these eggs, I would transfer the worm, the hermaphroditic worm, onto a new plate, wait a couple days to see if the offspring grew up, and once they did, we would count them and record. And we would keep going until the hermaphrodites stopped reproducing. And then after that, we still looked at the worm daily until it died, and then we recorded how long it had lived for. And we did 10 worms per bacterium, 1 worm per plate, so there was 150 altogether, and these were all genetically identical worms. These were our results. Here on the x-axis are the different bacterial species, and this here is the control on the far right. And on this side is the number of offspring, the average number of offspring the hermaphrodites produced on each bacteria. And what you can see in comparing the control is that these different clusters right here were significantly higher than the control group even. And that was cool because we could see that these worms actually liked these natural soil bacteria even more than the OP50 E. coli. And also we saw that these down here were pathogenic, and the worms could not reproduce on them as well as these other ones. These are our preliminary longevity results, and as you can see here, these are the 2 potential pathogenic bacteria, and they did not live for very long at all. In that, you can see the similarities between the number of offspring they reproduced and also how long they lived. It was very much a similar correlation there. And then also these are kind of clumped together. You can see this, they were on the other side before. So. My third question and final question was, can worms distinguish and choose between different bacteria? Now in humans, we all have to decide whether to choose the healthy and savory option, or the unhealthy, and then choose between a healthy and kind of bland option. And in the worms, we predicted, we did a food preference assay where we measured if they chose between 2 different bacteria on a plate. And what we did was we placed 1 of the 14 isolated bacteria and on one side, and then E. coli on the other, and we placed the worms in the middle and waited an hour, and we counted to see how many had traveled here, and recorded,